Hi, my name is Elizabeth Mateo Pagan. Welcome to my poster. In this study, we look the impact of the synthetic food additive, tert-Butylhydroquinone on B cell activation and antibody production. So B cells are part of the immune system that produce antibodies after being activated by T cells in the lymph nodes. The synthetic food TBHQ is typically used to preserve foods like cereals, cooking oils, and give them a longer shelf life. TBHQ also activates the transcription factor in NF2, which implicated in the stress response of cells. We have previously shown the NF2 activation by TBHQ can change the way B cells and T cells act under certain conditions. However, we have to look at this physiological interaction between B cells and T cells, which why we need in this study. In this study, we took two mice. One was a perfectly normal C57 black 6 mile. The other one was a mouse that is identical, but it don't have the NF2 transcription factor. Surgically remove the spleen from these mice and extract our cells from them. These cells will be mostly B cells and T cells because that is what is a spleen. We did activated our cells with a escalating dose of TBHQ ranging from 0.25 micromolar to 5 micromolar. We can activate them with an antibody cocktail that mimics a T cell activating a B cell. The antibody cocktail consists of anti-IgM antibody, anti-CD40, and IL-4. After 48 hours, we hurt ourselves and use flow cytometry to assess their activation and antibody function. In the first graph, we can see CD69. Here we have 6 different groups. In the background, no type of substance was added, unlike the others that TBHQ was added and the activation cocktail. With the presence of NF2, we see that it's faster. It's for that early activation marker CD69 is increased in NF2 deficient B cells at 48 hours. In the second graph, you can see the CD86 is a marker similar to CD69, but with a different function. They are co-stimulatory proteins. They activate T cells, but with the absence of NF2, we see that is lower. That is for delayed activation. In the graph of CD40 is a T cell communicator and co-stimulatory protein which requires activation. In this graph, there was no effect with TBHQ. However, we can see that there was in the genotype. The other graph, this we're talking about IgM. IgM antibodies are produced as a body's first response to a new infection or to a new non-self-antigen providing short them. Here we can see it on show a genotype or TBHQ effect. In summary, we observe that CD69 increase in NF2 deficient B cells at 48 hours of incubation, unlike CD86, which was decreased in NF2 deficient B cells at 48 hours. Although the 48 hours of incubation were good, there was no effect of TBHQ or genotypes in the IgM after the same as before mentioned. It should be noted that LPS was also used for activation. Unlike the antibody cocktail, it was like a gram-negative bacterium. That is why the absence of NF2 delays the activation of B cells.